This may be your next book, but could you say something about the gay brain?
Okay, so you may turn straight to Appendix 1 that's called The Male Brain and Sexual Orientation, where I fought with my editors and publisher to allow me to put that section in the book because I felt it needed to be there. If you're going to have a book called The Male Brain, you need to include at least something about all sexual orientations. Now, basically, the literature in science seems to indicate at this point that genetic differences at birth are probably responsible for what's called same-sex attraction. It's not some kind of a moral decision. It's not something that happens to you that changes you. It has to do probably with some type of brain circuitry that happens because of both genes and some hormones. For example, when teen boys that are straight start to go through puberty, they start to be attracted to female body shapes. They may see everything that walks by that looks like a pair of breasts. Straight guys are interested in, but at the same time, guys that are same-sex attracted are interested in guys. As a matter of fact, one very interesting study showed that males who are gay actually find they don't change their ability to smell male pheromones. They continue to be able to smell male pheromones and actually enjoy that smell much more than the female pheromone smells, which the straight guys like. So there's something that's going on at a very biological level. So thank you for that question. We need a lot more research before we're able to say much more definitive about that area. But thank you.
Humans have 23 pairs of chromosomes. One of these pairs controls sexual determination in humans, the X and Y chromosomes.
Individuals with two X chromosomes are female. Individuals with one X and one Y chromosome are male. These chromosomes also control the strongest genetically determined behavioral trait in humans, sexual preference.
In most cases, individuals with a Y chromosome, i.e. males, are attracted to females. Individuals with no Y chromosome, i.e. females, are attracted to males.
This is how a human gets made. Every one of us started out this way. We've a pretty good idea what happens over the 9 months it takes to produce a newborn. But there's this one bit that's been missing. Right at the beginning, before this animation even starts. The very first few weeks. Now armed with new ways of growing human embryos in the lab, scientists are learning what the very beginning of human development looks like. For decades, studying this crucial early time in a human embryo's development was a technical headache. Embryos are hard to get and keep alive outside the body. And it's an ethical challenge. Guidelines put together in the late 70s and 80s prevent scientists from growing embryos for longer than 14 days. Despite the hurdles, scientists do know a bit about what happens in those critical weeks, often from animal studies or rare human tissue samples. They know that sperm fertilizes egg, and one cell grows to 2, 4, 8, and so on. Around day 5 or 6, the blob starts to feature different types of cell. Scientists call this blob a blastocyst. All being well, the cells in the blastocyst begin to differentiate.
The single cell has transformed into a complex network of over 200 different types of cells.
1 week in, it implants into the wall of the uterus. What happens next has been a bit of a mystery. By implanting itself into the womb's wall, the embryo basically hides, and that makes studying it in humans impossible. One way of exploring this crucial time is to study embryos donated by people who no longer need them for fertility treatment. In the last few years, several labs have developed new ways to nurture these embryos. Their techniques have allowed them to start building a picture of human development that's more detailed than ever before. Here's a day 6 embryo growing in a lab. Cells destined to become the actual fetus are tagged in green. Those that go on to form the placenta in blue. By day 8, the cells are sorting and arranging themselves. The green fetal cells have condensed together, and in red here are cells that will form the interface between the baby and the placenta. It seems these extra embryonic structures develop a little later in humans than expected from studies in other animals. Day 10. The whole thing increases in size, and bundles of cells start developing into support structures. Surprisingly, at this stage, the embryo can direct its own development with no input from the mother's tissues. By day 12, the outskirts of the embryo are preparing to bind more strongly to the wall of the uterus. After all, if this were a natural pregnancy, the embryo would be there for the next 9 months. You can even see little holes appearing, ready for the mother's blood vessels to start supplying the embryo with essential oxygen and nutrients. By now, the embryo is signalling its presence to the mother via a hormone, the one that pregnancy tests pick up, HCG, labelled here in yellow. After 2 weeks, the teams ended their experiments in line with the ethical 14-day limit. To study what happens after 14 days, researchers had to turn to different techniques. Recently, scientists have built artificial embryo-like structures from stem cells. Using these partial models, they can study things like cell signalling, or even the formation of the primitive streak, the crucial thread of cells which guides a process called gastrulation. That's the moment the embryo decides which end will become the head. Here they use human stem cells growing in an animal embryo to explore that process. But even after this early phase is over, there is still a lot of work involved in building a body. To study later phases, scientists have made and analysed high-resolution 3D atlases of human embryos and fetuses. One team found that the left and right hands don't simply mirror each other when growing their nerves. Instead, some branches take random paths in each limb. Another group saw muscles that grew in early embryos only to disappear as the fetus developed, like these.
But how did this complexity occur? The key to understanding this transformation is found within the nucleus of our cells, where our genetic material is stored. Our genetic material is made of both our genome and our epigenome. The genome is the complete set of DNA found in the cell. Each cell in our bodies contains 2 copies of 23 DNA strands, one from mom and one from dad. Each strand of DNA consists of thousands of segments called genes, which encode for and regulate protein production. Proteins are large molecules that perform the majority of functions in our cells. To regulate protein production, each of the 46 DNA strands are coiled around histones, much like thread wrapped around a spool. The epigenome consists of chemical tags that modify the genome and tell it when to turn a gene on and when to turn a gene off. When a gene is turned on, it unwinds from the histone so that the instructions for protein production can be read by a group of proteins known as the transcription factor. If a gene is turned off, the gene remains tightly wrapped around the histone and is inactive. Unlike the genome, our epigenome changes throughout our lives by adapting to environmental cues. As our cells divide and differentiate, they become more specialized by altering their epigenome. For instance, genes turned on in muscle cells may be turned off in skin cells. Therefore, the epigenome of a muscle cell is different from that of a skin cell. As we grow and develop throughout our lives, our epigenome continues to change based on lifestyle and environmental factors. What you eat and drink, the medicines you take, and the pollutants you encounter can all impact your epigenome.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
It's becoming ever clearer just how important a human embryo's first few weeks are. Many scientists hope that more research will lead to a better grasp of why some pregnancies fail and how birth defects arise. Maybe even make in vitro fertilization work better. But some just want to understand exactly what happens to this tiny ball of cells in this short spark of time at the beginning of all of us.
In their first few weeks, all fetuses develop along similar lines. If nothing changed, we would all be born female. Fetuses with the male Y chromosome will form testes at about week 6 that begin to produce the hormone testosterone. But at about the 8th week, testosterone is released and may affect early brain development. This hormone masculinizes the body. Testosterone also masculinizes the brain, including a part called the hypothalamus, part of the network which controls who we find sexually attractive. Some scientists believe that the more the hypothalamus is exposed to testosterone, the more it sets the stage for a sexual inclination towards women.
The prenatal environment can influence sexual preference. Hormones have an important effect on human sexual orientation. They determine our sexuality while we are still developing in the womb, through exposure to sex steroids. For instance, an exposure to a lower concentration of testosterone would lead to a female-typical orientation.
But we have to keep in mind that sexual preference is innate, and the idea that someone's childhood or cultural or social factors can determine their sexuality is untrue.
Epigenetics is sort of a revolution in the biological sciences, and what epigenetics refers to is sort of a layer over your conventional genetics. So it's sort of this almost like a circuit board that's able to turn on and off genes. And what happens is that genes can essentially be permanently turned off for the entire lifetime of the organism, and that can persist for multiple generations. So in our case, we're looking at plants, and in the case of plants, it's not really understood why plants flower in the springtime or how they know when to flower. And so it turns out that there are epigenetic switches that turn on genes for flowering just at the right time and just at the right moment. And these molecules we think are RNA molecules, which are DNA's molecular cousin. This is a new class of RNA called long non-coding RNA, sort of long junk RNAs. And these are 100 to 1,000 times longer than most of the RNAs that are normally studied. And in our case, we can show that they actually have quite detailed structure. Much of our genome and also plants' genomes used to be thought to be junk, so-called junk DNA. First of all, they were thought to do almost nothing. But in the last 5 to 10 years, it's been shown that a lot of this junk DNA actually does a lot of stuff. And in our case, the junk DNA codes for RNA molecules, and these RNA molecules can actually do things. In our case, they trigger the plant to flower at just the right time. Also, these switch states can be passed down to future generations.
80 to 100,000 chemicals in use in the United States today, depending on where you look. Among those, there's a number, somewhere around 3,000 according to the EPA, that are considered high production volume chemicals. These are chemicals that are produced or imported into the U.S. at volumes of greater than or equal to 1 million pounds per year. First example is frogs. This is the work of Tyrone Hayes out at Berkeley, who in the early part of this century put out a series of papers showing that the pesticide atrazine was leading to demasculinization and hermaphroditism among frogs. So they dosed these frogs with the atrazine at low doses, and they were finding that they were growing both, forming both male and female reproductive organs. Another chemical many of you are probably aware of, this is phthalates. In the late 90s, several different groups were reporting male reproductive tract anomalies in rodents dosed with very high levels of certain phthalates.
In 1998, University of California professor Tyrone Hayes was invited to join a panel of scientists to study the effects of the herbicide atrazine on frogs, fish, and other animals.
Atrazine is an herbicide, and it is, in a nutshell, it is the largest selling agrochemical in the world. It is the top selling product for the largest agrochemical company in the world. It is used on the top crop for the largest economy in the world, the U.S. And one of the things it's used to fight is the most common weed in the world. So it's a big deal. And one of the things that we know now, and this is being shown all over the world, is that atrazine's everywhere. It's ubiquitous. I just came back from a meeting of chemists talking about atrazine and how it sticks around for 10, 15 years. I mean, my biggest concern would be the water. If you live in the Midwest and every bit of water you drink is contaminated with atrazine, your rainwater. I mean, imagine. Rainwater has enough atrazine in it to make hermaphroditic frogs. Atrazine caused males to lose their androgen, their testosterone. The result is these males become chemically castrated. In some cases, they'll completely grow ovaries mixed in with their testes. In other cases, they'll grow eggs and actually yolk eggs inside their testes instead of making sperm. What does it have to do with humans? Humans don't grow eggs in their testes. Well, but humans get breast cancer. And the same gene, it's involved in breast cancer. The hormone that's involved is exactly the same in frogs as it is in humans. 1 million people a day are exposed to atrazine. I think it's not just frogs, it's fish we have to be concerned about. And it's not just fish and frogs, it's how they fit into ecosystems. A little frog in a fish bowl is not all that different from a little fetus in the womb. I mean, our fetuses, humans, live in an aquatic environment just like amphibians, where they're breathing in and sucking up and soaking themselves in the water for 9 months, just like our tadpoles in those little buckets. And many of those chemicals, our placenta aren't designed to filter out these synthetic chemicals. Many of these chemicals are in that amniotic fluid. We know that now. And then you also have to be concerned about not just you and I, but the farm workers that are exposed to huge levels, that are applying this stuff.
Atrazine is used to control weeds in cornfields, sugarcane fields, residential lawns, Christmas tree farms, and golf courses. In 2003, after 10 years of contentious scientific debate, the Environmental Protection Agency reapproved the use of atrazine in the United States.
Syngenta itself, which used to be Novartis, used to be Ciba, of Ciba Geigy. It's a Swiss-based company. The reason I laugh is atrazine's never been legal in Switzerland, ever. So here we have a company in a country that makes a compound that has never been legal in their country. They sell it to us and we expose 1 million people. We use 60, 80 million pounds of it a year. It's banned all over Europe now. But they'll sell it 80 million pounds of it a year, $500 million a year of it to the US. And we'll spread it all over the place. What's important for this talk, however, is that the effects that we're talking about now are in adults. When we start to talk about early development, whether we're talking about amphibians or humans, the effects are much greater. What is a little bit of poison in an adult is a great amount of poison in a developing infant or even in a developing fetus. What's interesting now is there's discussions that women who breastfeed might have lower risk of breast cancer. And some of the discussions are that those lower risks might be because women who are breastfeeding are mobilizing those toxins from the breast by excreting it into the milk. But those toxins then, which are in the milk, are going into the developing infant. And the difficulty that we're faced with now, as Gina alluded to, is it's one thing to show an acute exposure and a problem, but much more difficult in humans to show that if I get breast cancer or prostate cancer at 50, that it's related to something that I was exposed to from my mother's milk, or it's related to something that I was exposed to in the uterus that came across the placenta. What's been very helpful in these areas are that we have rodent studies. We have rat studies, which they're mammals like us, they breastfeed like us, they carry their babies like us. And we're now seeing that, for example, atrazine exposure in rats leads to prostate cancer in the females when they're nursing and when they're pregnant, so it crosses a generation. We're seeing that atrazine causes hormone imbalances that lead to breast development problems in the daughters of rats that are exposed, and then those rats even have problems feeding their daughters because their breasts don't develop properly. So we're seeing in rats that we get problems in the grandchildren of exposed rats, 2 generations down. So the affected rat who has poor neural development and poor growth was never exposed to atrazine, his or her grandmother was. And what you'll often hear from the industry is, well, rats aren't people. I even saw this on Channel 7 just ran a news story, and there was a guy at Stanford saying, well, mice aren't men. But we've seen this already. We have a whole history. DES, which we now know DES causes vaginal cancers in the daughters of women who took it when they were pregnant. We know that DES, or diacetylstilbestrol, causes uterine malformations in humans when the mothers were exposed, but we'd already shown that in rats. You can expose a rat and 1, 2, 3 generations down, you still see effects of DES. We already knew that in DDT it could cross the placenta and affect developing rats. And now in 2006, finally we have a study showing that women who are exposed to DDT before the age of 14 are more likely to develop breast cancer when they're 50 or 60 years old. So we already know that these rat models, and of course the other irony is that these companies use rat models when they're trying to get the chemicals on the market. But then when you find something negative, they argue, oh, it's just rats, it's just mice, it's not relative to humans. So my concern is raised for environmental health, because now we're seeing a global decline in amphibians, and I firmly believe that pesticides are playing a role in that decline, not the sole reason, but playing a role. And I firmly believe that what we're seeing in amphibians and what we can model in laboratory rodents is a warning about public health issues, because many of the developmental processes, many of the hormones are the same. And in particular, I think we need to be concerned about, I've often said with the atrazine issue that we need to take a stand, write to the EPA, write to Congress, and be proactive, not because of us. Essentially, we've already been exposed. And then when you're given the half-life of many of these chemicals, our children have already been exposed, and our grandchildren will likely be exposed. So the fight we're fighting now is really, if you look at what we're learning from rats, the fight we're fighting now is really for our grandchildren's grandchildren.